Hi guys, today class we are going to discuss about the most important topic in medical instrumentation, that is the clinical significance of EEG. So before starting that we should know what is EEG. So EEG is nothing but is a study of electrical activity of the brain as like a ECG, which is a study of electrical activity of the heart. So here the electrical activity in the sense, so here we are measuring the summation of action potential which is generated by the neurons, that is the millions of neurons which is present in the brain. So by analyzing those action potential, we come to know what is the function of the brain. So that is all about what is EEG. Yes, so to pick up these brain waves, that is action potential, we need something called electrodes. So already you know that for measuring ECG activity, we need some electrodes, that is surface electrodes like that. So here also we need electrodes to measure or pick up the brain waves, that is EEG electrodes. So there is usually the two methods to pick up the or place the electrodes. One is non-invasive method, another one is invasive method. So if you take invasive method, we will have a direct contact with the targeted area. Here in this case, if you are measuring the brain activity, brain electrical activity, in the sense we will make a direct contact with the brain. So as doing this, we can able to measure the action potential of the particular area, right? So if you are measuring from the direct if you are measuring by means of direct contact, that is from the surface of the brain, we will have the action potential which is in the range of 10 millivolt. So whereas in the opposite case, that is a non-invasive method, that is we will place electrodes just over the skull, that is over the surface of the particular area. We will have the output in the range of 1 to 100 millivolts. So these are the output which we will get either a non-invasive method or invasive method. By analyzing this output, that is the action potential, we can have information about what is the function of the brain activity, right? So again, this action potential usually varies with respect to where we are placing the electrodes, whether it is inside the skull or over the skull, right? So next we will see the origin of EEG. So that is the source of EEG. Mainly we speaks about EEG is nothing but electrical activity of brain, that is the action potential. But here this case, unlike ECG, here we will measure only the average, that is a summation of action potential where the millions of neurons being involved, right? So if you take ECG, we can easily tap the signal from the particular cell of any muscles cell, right? But whereas in EEG, it's very difficult for us to measure the action potential of the particular neurons. So we can only measure the graded potential, which is nothing but average of action potential, that is average of resting potential. So whenever we speak about EEG, we can measure only the graded potential, not like ECG, right? So these graded potential usually found in two forms. One is inhibitory post-synaptic potential, another one is excited post-synaptic potential. So we know the output of the EEG is it's a waveforms. So usually if you take any waveforms, you have both positive and negative direction. So based on the movement, that is the direction where the waveform is moves towards the threshold, there are two types. One is EPSP, another one is IEPSP. As I told you before, EPSP is the excited post-synaptic potential, IPSP is the inhibitory post-synaptic potential. So the ESP is the potential which is increased in positive direction, that is more close to the threshold. If you see the IPSP is the again that is the opposite direction, that is when the membrane potential while measuring it, when it moves towards the negative direction, then it is IPSP. So these are the main two type of potential if you see in graded potential, right? So next we will brush up the few basic things about anatomy of our brain. Already we might have studied in our lower classes. So these are the main parts like cranium, cortex, brainstem, spinal cord, cerebellum, dura, and la ganglia. So these are the main things we should know before we getting into EEG. So that's why I'm teaching what is basic of anatomy of the brain. So another most important things we also more concerned when we are measuring EEG, that is the lobes. We have basically have four lobes: occipital lobe, parietal lobe, and frontal lobe, and temporal. So here I have highlighted with the colors. Okay, so to differentiate the different types of lobes. So these four lobes will have each specific roles are controlling action over our body. So if you take frontal lobe, which is mainly involved in problem solving, emotional traits, reasoning, speaking, and voluntary motor activity. If you take parental lobe, again it plays a different role like reading, body orientation. If you take occipital lobe, it has like a vision and color perception. And again, if you take temporal lobe, it mainly involves in understanding the language and behavior and memory of particular person and hearing capacity, right? So let's we will see what are the basic five different types of brain waves which usually we will get when we are recording EEG, right? So these are the five basic brain waves. If you see the brain waves, it is usually irregular pattern. We don't have a standard waveforms as like a ECG and EMG. We have a standard waveform, right? But in case of EEG, we don't have a standard waveform pattern because we know that the brain are very much involved in so many actions. It's so much connected with the emotions. So the brain waves will keep on vary time to time, moment to moment. So it is very hard to pick a significant standard waveforms. So, but whereas we will have the classification of brain waves based on the frequency. So if you see the frequency range, it starts from 0.5, it ends with 100 hertz. So we'll have different types based on this classification. One is delta, theta, alpha, beta, and gamma. To be short, we will have some trick to remember this, that is D tab G. So D is the delta, T is the theta, and A is the alpha, and B is the beta, and G is the gamma. So that is a D tab G. So as D is the delta, that is the lowest frequency range it covers. So one starting from 0.5 to 4. Then followed by we have theta, that is 4 to 8, and alpha 8 to 13, and beta 13 to 32, and gamma 32 to 100. So each and specific brain waves will represent some functions, that is the status of the person. So if you see the delta, that is a 0.5 to 4, it mainly represents the status of the particular person where that is the person has undergone a deep sleep, or they don't have a conscious at all, like that. So if you have the delta waveforms, it represents the person having deep sleep or loss of consciousness. And if you measure the brain form of some person who is having light sleep and reduced consciousness, means that at the case we will get the theta, that is the frequency range of 4 to 8. Similarly for the alpha, if you are getting these type of waveforms when we are measuring EEG, in the sense that it means that the person is so relaxed both physically and mentally, so without any disturbance like that. For if you take beta, that is 13 to 32 hertz, it means that the person is so awake and alert, that is they are very much concentration of particular things what they are doing, or what they are dealing with that. So in that case the person is like having beta waveforms. First at last, if you see the gamma, that is 32 to 100 hertz, if you are getting these type of waveform, in the sense the person is very hyperactive. They are very much in concentrated to the particular work what they are loving to do, and as well as they are very much focused, especially when they are problem solving some critical problems or doing some critical task, like that. So based on that emotional tie-up, or based on that status of the brain, we have the different waveforms. All these waveforms are mainly characterized by different types of frequency. So by measuring or analyzing this waveform, we easily come to know what is the status of the brain of the particular persons. So even we have options of identifying whether the person is telling or telling lie or truth, or whether the person is so anxiety, or they are feeling so bad, panic, or they are crying, or they are so happy, like that. So we can easily identify what type of the person, what type of the emotion the person is like that. So the EEG is like is a vast area. We can have so much research to do, because the brain is like it's a control processing of a body, which involved in all controlling actions, so even right from emotions and the voluntary involuntary actions, memory, everything. So this is like a ocean. We can do a number of research related to our whatever we feel, right? So that's all about today class. In this class we will see about what is the electrode placement of which is being used to measure EEG. That is a 1020 electrode system of EEG. Yes, thank you guys.